Hi, my name is Mark Muller, and I'm the CEO for Topogen Incorporated. I'm going to present to you a video that describes our new DNA repair systems that we've developed. Now, these are cell-based DNA repair systems that you can use to interrogate either homologous recombination or non-homologous end joining. So let's get started. In this particular video, I'm going to talk about the HR system specifically. I will mention non-homologous end joining in passing. We have another video that focuses on the NHEJ pathway, and you can consult our website for this information. The overview of this entire system for both HR and NHEJ involves starting with an engineered cell that's GFP negative but still has the GFP reporter, and this particular gene is inactivated usually through a mutation. So the cell is negative for the fluorochrome. And what happens is we introduce a double-strand break, and this shows an example of using a homing endonuclease called SEE1, but you can use any system for making the break, as long as the break is introduced in a very specific way. Now, these particular cells are engineered with reporters that will break specifically within the GFP locus. However, you could introduce DNA breaks with guide RNAs using CRISPR-Cas as well, and we have products that will do that. So we encourage you to look deeply into this aspect for your specific system and consult with us, and we can advise you on how to design your cell lines for repair. Now, what actually is going on here is the double-strand break is being introduced by ISCE1, and HR will ensue over a period of 2 to 4 days. Now, the signal strength, that is the GFP positive character as a result of HR, will gradually increase and plateau up to about 5 to 7 days, but you'll start picking it up in 2 days, and I'll show you some data on this as well. Of course, early on, you're going to just see a few of these GFP positive cells, and these are called progenitor cells. These are parentals post-HR, and I'll describe that in more detail in this video. So here's some of the cell-based kits we currently have, and these are available for sale through our website, and pricing and availability can be found at topogen.com. So we have, first of all, human cell lines that will report both HR and NHEJ. We have a number of different HeLa cell systems. We have neuroblastomas, so we have lineage-specific lines. We also have some mouse cell lines for HR and NHEJ, and those are great for people that work in mouse models. That said, you can go to your system if you can establish a cell line, a particular cell line you're interested in a mouse line, for example, or in rodent, then you can send us those cells and we will engineer them for you. This is part of a custom service, which I describe next. We can engineer your cells to host HR and HEJ, and this is a service that takes a little bit of time, but we have great experience in this arena. So we can do disease-specific model cell lines, iPSCs to model cell lines. It's a big deal these days trying to develop systems to look at disease at the single cell level, for example. Finally, we can engineer any lineage-specific lines as long as we can grow the cultures. You know, the cells show continuous growth and are immortalized. So this is something that may be of value to those that are interested in DNA damage, DNA repair pathways. Now, Topogen is a company that focuses on topoisomerases, as you may know. However, we're opening up this area because topoisomerase poisons specifically cause DNA damage, and so we're interested in developing models to look at DNA damage by topoisomerase active agents as well. So this is a new line of products will be coming out in the next quarter. Now, what are the applications for these kits? What can you do with these things? Many people will ask, what are the tools available to us? And we believe that these kits are extremely valuable for looking at events that are physiologically relevant at the cell level. I think gone are the days where you can do isolated biochemical experiments and really get good, strong physiological feedback. Whereas if you install a clever reporter system at the cellular level, the potential is much, much greater that you're going to get something useful. So let's talk about these briefly. So if you're a lab and you're working on mechanisms of DNA repair and you're doing knockdowns, gene additions, bringing in novel factors, these kits will be very, very valuable to you, with the added advantage that you can install these reporters in virtually any cell line. If you're looking at pathway proteins, you're looking at signaling, you're looking at genes that are important as support structures for DNA repair. If you're interested in finding out what are the decision points for going with HR or NHEJ, those kinds of questions are very answerable with our system. So we encourage you to try them out and see if it's going to work. DNA repair events in the cell cycle are also approachable experiments with our kits. Again, these are cell-based, so if you manipulate the cell cycle, if you analyze things and synchronize cells, this has value. There's evidence that in aging, our DNA repair mechanisms decay as we age, so this appears to be related to NAD metabolism, but these experiments recently reported in Science were worked out using these kinds of DNA repair cell-based kits. So looking at in situ DNA repair in aging research is a popular area. Another aspect would be finding druggable targets. What's out there for drug discovery? We can find small molecules, small effectors, natural products, synthetics. What things can inhibit repair? Obviously, this is important in growth control. It's important in apoptosis. It's related to cancer. So cancer research labs may find this useful. Cancer cells are frequently deficient in DNA repair. Can that be exploited in order to eliminate tumor cells? Another valuable contribution for these kits. Chemical library screening. This is where you have obviously a large number of compounds, synthetic compounds that you want to test. We believe that even though this technology we describe is not really high throughput, you can create a high throughput system by going to a plate reader and just reading GFP output. And this can be done with the TCAN unit, for example, or other technologies that currently exist. So chemical library screening is HTS, I would call it semi-HTS, would be something of value for this kind of kit. So chemists, for example, may be interested in this. Now let's move in on the HR system and talk specifically about homologous recombination. Homologous recombination is also referred to as gene conversion. These two terms are used interchangeably. Without drilling down on the details of HR, let's just give a couple of precepts. With HR, you recover a sequence by homology to another sequence of the genome. So it's necessary you have an exact copy somewhere else. This is accomplished through a process called synapse, where the cell has the ability to find a homologous sequence, typically in a sister chromatid. This tends to be a highly error-free process and is cell cycle limited, typically in S to G2 phase. Now these are conditions where you have a sister chromatid. Obviously the amount of DNA is doubled because they've gone through S phase, but they haven't divided yet. So a very robust system, not all that common in animal cells and eukaryotes. I think the predominant repair pathway would be NHEJ, but however, this is an important system. And you can approach any kind of studies using in situ screens with cells to look specifically at an HR pathway. Very powerful technology. So let's talk just a few minutes about the details of how this works. Now this is just one example of a GFP reporter that we would use. Now as I said, we installed this stable reporter that has a cassette of GFP, cassette 1, entire GFP gene, but engineered with some kind of damage, some kind of mutation. In this case, it's a stop codon, and there's a homing endonuclease called SCE1 that's co-located near this. And then downstream 3', we have this cassette 2, which is wild-type donor sequence. Now, the wild-type donor sequence then has the ability to synapse back after a double-strand break and repair this region back to wild-type. So the question is, you have to introduce, how do you introduce the homing endonuclease, SCE1? Well, there's two ways you can do it. We have a simple method for transfection, and these days with nucleofector or with standard transfection, you can get very good transfection efficiencies. Typically with nucleofection, we see transfection efficiencies of in the 60 to 70% range. And so that's what's going to happen there is the SCE1 is going to come in and make that double-strand break, and that's going to convert, or gene conversion or HR will happen and your cells turn green. We also have systems where we can put the DNA damaging protein on a TET-on promoter. And this is really nice because now you can just add back DOX, and DOX will induce SCE1 if we're using an SCE1 damaging mechanism. And this will lead to double-strand break and repair, and the cells will turn green. And you can very easily mark this by just looking at the cells and looking for green cells. Now, it's important to note that even in the absence of SCE1 or in the absence of DNA damage in this system, there's always going to be a few cells that are GFP positive. And typically it's low, 0.1 to maybe as much as 0.5%, depending on the cell clone that we choose. However, you can distinguish between the real HR process and a background cell by doing live image. And I'm going to try and demonstrate that to you in just a minute. Because in live imaging, if you do a time lapse, you would see after addition of SCE1, you could track one cell, for example, this cell, and it turns green while you're looking at it. That means that that cell must have been damaged by SCE1 in this case, and that led to a DNA repair event, or HR event in this case. Now the reason we call this a dedicated HR reporter is because we have this wild-type donor sequence down here. So the wild-type donor sequence is what defines homologous recombination. So in this system for HR, but it also applies to the other pathway, NHEJ, we use a readout based on GFP percentage of cells, the presence of these progenitor cells, which I'll demonstrate in a moment, and also GFP intensity. These can be measured by imaging, by live imaging, by flow cytometry, imaging cytometry, or even using like a plate reader to measure total GFP fluorescence. So this is very robust and a very tractable system for looking at the HR pathway in cells. Now I just want to show you a little bit of data demonstrating that this is real. And what we've done here is we've just taken some HeLa cells that have one of our reporters, and we just did some immunofluorescence labeling with phosphorylated gamma H2AX. Now this is a protein that's going to mark sites of DNA damage, and it's very common in the DNA repair field. Now look what happens when we add SCE1. You can see these foci appear. So this is telling us that the SCE1 is in fact doing its job, introducing double-strand breaks and causing DNA damage. But more telling is when you look at the scatter plots from a FACS analysis from flow cytometry, you can see that the GFP positive region up here has very few cells in the absence of SCE1, but when you add SCE1, there's a large uptick in the GFP positive character. These cells would be autofluorescing or dead cells. So we can rule those out. And it's very important in these experiments that you always do a control where there's no SCE1, because you must establish your background. And you can see that in a simple histogram here that after 48 hours, in the absence of SCE1, there's essentially no GFP. And now we're up to about 5.5% GFP at this particular juncture in the experiment. So that's telling you that, oh yeah, you're getting GFP positive cells and the system is working as expected. Now, this is where the real power of the technology, we believe, comes to the forefront. This is a new field, and we're going to call it opto-DNA repair. Now, this is using optics to analyze DNA repair, and we're adding dimensionality to this analysis. In other words, we're adding a time dimension in order to analyze the repair process, and you can do this down to the single cell level, which we believe is the ultimate in resolution. So let's take a look at a live imaging of HR in cells in this experiment. So what you're looking at here is these progenitor cells coming online. Here's some cells here. Here's a couple more here. And these cells go from no fluorescence to being positively fluorescent, as you can see in this experiment. That means that those cells are specifically damaged by SCE1. Gene conversion occurs and the cells turn green. This is very obvious in this experiment, and you can actually enumerate the number of progenitor cells if you want to get quantitative data out of analysis like this. This is happening over a period, I think, of about 4 days. We're looking at these cells. Now, in the control cells, that is cells that have not been treated with SCE1, there's been no DNA damage, you may see the occasional green cell, but you won't see progenitor cells. You'll see, an occasional clone that's got a few green cells, but they don't come on as such in live imaging. They're pre-existing, so we know those are background cells, and we can ignore those. So thank you for listening to this video, this screencast. In order to inquire about any of our new products, these new DNA repair kits, I encourage you to go to our website. We have a number of these in production. They're being developed now, including multiplex systems, which allows for interrogation of both DNA repair pathways in the same cell. In other words, you can look at HR and NHEJ with different fluorochromes in the same cell. So you can study the pathway decisions that are important in defining which pathway is going to be operable. And it might be possible to select drugs that affect one pathway or the other. So this has real value in future research. If you would like to investigate further, please contact me directly through support@topogen.com. And we have a history of offering strong technical support for our products. We provide free consulting. When you buy our products, we don't just cut you loose. We work with you. You can contact us, you can show us your data, we'll help you with the analysis. And our goal is to provide tools for this research and then help you enable those tools so that you can use the tools in the most appropriate way and get good publication quality data out of the analysis. Everybody has different instrumentation in their lab. So to some extent, your platform for fluorescent activated cell sorting or cytometry or imaging may be a little bit different than ours, but we will work with you in an attempt to adapt and technology transfer what we have in our inventory into your lab to help your project advance. And again, thank you for your time.